Next we talk about pleiotropic or pleiotropic genes. Now what are pleiotropic genes? Here certain genes, there we read that there are so many alleles for a single gene. Now here we are talking about a single gene having that particular gene controls more than 2 characters. Now, for example, sickle cell anemia and phenylketonuria. Now we're starting about linkage. That what is linkage? Linkage is discovered by Morgan. He can't able to tell about crossing over but tell about linkage, and similarly Mendel can't able to tell about linkage. Now what is linkage? If we tell what is linkage, then the distance between the genes is directly proportional to recombination, and it is inversely proportional to linkage. If we go through this formula, then we can see that now this is a chromosome where allele A and gene B. So here the distance between the gene is more. Now here the chances are more of recombination. So what will happen over here? There will be a recombination takes place. Small a and capital B and capital A and small. Then the sequence is transferred from one parent to the next parent, other from one chromosome to next chromosome. Now if we talk about linkage, here A and B are the genes. Now the distance between the gene is very less. So if the distance between the gene is less, then the linkage will be more. So the chances of linkage will be more in that part and recombination will be less. Now what we can say that A, B and B. Now what is linked gene? The distance between the gene, if the distance between the gene is very less and they are linked with each other, that are called linked genes, and the linked genes, they as it is move from one place to another without separating from each other, that is called linkage, and the other one is called recombination. Example of complete linkage is Drosophila. Incomplete linkage red-eyed Drosophila when crossed with wild type Drosophila creates incomplete linkage. Next is determination 6. If we talk about humans, we know that In humans, the female is homologous with XX and the male is heterozygous with XY. But we've talked about birth, that is cow, you can there. The female is heterozygous ZW and the male is homozygous with Z. Next, when we talk about the housefly, here the female is again homozygous and the male is heterozygous. Last, if we talk about butterfly, here the female is and the male is in grasshopper. And lastly, now we in pedigree analysis helps to detect the ancestor and the line of a particular ancestor. And some of the rules: the square-shaped color is called diseased male. A circle, fully colored circle, this condition is called carrier. Carrier for the disease, where the condition is, for example, this is the carrier where the disease is only on the one allele, not the other. And the disease, if we talk about disease, the disease is allele is both the allele are diseased. Here, single allele is diseased. Similarly, this condition or this condition of this condition is called carrier female. Again, single allele is unspecified sex. Multiplication mating between relatives. Consanguineous mating between the relatives. These are the children. This is the H1 generation. You can see child, and this is the parent. This 5 unaffected individuals for offspring. This is the condition of deceased or twins. Including those rules. Now, which are autosomal diseases and which are sex-linked? If we talk about autosomal diseases, either of the male or the female can be affected by any of the affected parents. That means we are saying that if or we can say that a affected father can produce a affected female or a affected male, and from a affected female, any of the parent, daughter or the son, both can be affected. Either of the one can be affected from the affected mother. But in the case of sex-linked, the affected male always produce a affected daughter, whereas a affected mother always produce a affected son who is dominant or recessive. We talk about dominant. No generation will be skipped. With one generation, we have a diseased female, and further we get, and this next generation we again get. So here, not the F1 generation, not the F2 generation, the disease is skipping, so that is a dominant. But if we talk about recessive, for example, here is a diseased female. Now whereas if F1 generation is having diseased, F2 generation no disease, and F3 generation is disease. Now here the one generation is skipped, that is called recessive. Solve one equation from that. Pedigree means we have to read it from bottom to top, because this is our generation, this is our ancestor's generation. If we look into that, here both the females are affected by a female. From this condition we can say that it is an autosomal disease. After that, both females are affected but either of the parents are not affected. Then again we can say that it could be carrier and maybe it is an autosomal disease. Now here, a female and a male affected by an affected female, so that is also, so this is confirmed that it is an autosomal disorder, because it is not confined that a female is affecting only the female or the male is affecting only the male. Now talking about the dominancy or recessive. Now the F1 generation is also affected, F2 generation is affected, and F3 generation is also affected. Now this is the autosomal dominant disorder. Now according to, by reading these rules, and this is how you can practice more and more questions to learn pedigree analysis, because it will be better if you practice more and more. Thank you.